Thank you. All right, so my last lecture is about gene expression, about genetics of gene expression, or expression quantitative trait locus analysis. So QTL, or quantitative trait locus, is a gene trait, measurable trait, which is associated with or linked to a region in the genome by some statistical method. So in classical linkage analysis or pedigrees and breeding analyses, it was just a portion of the genome that was co-segregating with the trait. So in an expression QTL then is a region of the genome that's associated with the expression, with a measure of gene expression. So gene expression is the trait, or in fact, more accurately, 20,000, the expression of each one of 20,000 genes, the parallel traits. So what we're doing in eQTL analysis is drawing up an association between the levels of transcript abundance in the tissue or cell type and all of the genotypes throughout the genome. And essentially, this can be done in a linkage framework in crosses, which is what it was done for the first 5 years or so of the discipline. But these days, more and more, we're talking about an association study framework. So the background to this is that the architecture of transcription helps to map phenotype onto genotype. So anybody who's just interested in genetic analysis, particularly by genome-wide association studies or linkage for that matter, is just looking for association, so correlation between genotype and phenotype. And we now know that about 90% probably of those associations are due to variants located in regulatory DNA. So in other words, only 10% at most are coding variants that affect the structure of the protein. So most things are regulatory. And so a large part of that genetic variation affecting complex traits must be due to genetic polymorphisms that affect some level RNA production or degradation or splicing or factors such as that, okay? So the idea then is that if we can measure the transcriptional phenotypes, then that's sort of a bridge from the molecular genotype and the DNA to the observable phenotype. So as I just mentioned, expression QTL are quantitative trait loci, that is regions of the genome associated with gene expression trait. In other words, they're thought to modulate transcript abundance. And they can either be measured in pedigrees, or classically they were measured in pedigrees or crosses. These days, we actually more commonly refer to things as e-SNPs as being SNPs that associate with transcript abundance, okay? So in a linkage framework, you're measuring a co-segregated portion of the genome, so that could be a large haplotype, whereas in GWAS, we're actually looking at individual SNPs. So these are the same thing, it's just that e-SNP is more refined and it's referred to the association study framework. Now, you can ask so given what I just said about architecture, the expectation is that genome-wide association study variants and expression SNPs ought to co-localize. Okay, so the reason being that if the e-SNP is responsible for the expression variation, and the expression variation is what's mediating the GWAS effect, then those e-SNPs ought to be the GWAS variants. And I'll show some data in a little bit from single-cell e-SNPs, so it's getting better and better. But even so, currently the best estimate of any published studies is that about 20% of GWAS variants actually can be localized with confidence to an e-SNP. So we're a long, long way from understanding why it's that complex. And probably a lot of the reason is because e-SNP effects are very cell type specific and in fact cell state specific. And it's quite likely that the effects are mediated at a snapshot in time. So for example, with lupus, maybe it's the case, at least one of my current hypotheses is that actually there's a specific phase of B cell development where GWAS variants tend to overlap with e-SNPs. And if you look outside that particular phase, if you don't actually do accurate enough typing of subtypes of B cells, you won't have the power to identify that co-localization because you're just not looking at the right cell type at the right time. Okay, so we've got a lot of work before us to sort of validate this paradigm that GWAS and e-SNPs are the same thing. I wrote this next sentence a few years back. It's kind of a silly sentence, I actually think. But anyway, if you just took two people, or you took two cows or two corn plants, and you just were able to repeatedly measure them, you probably find that at least 10% of the transcripts differ in abundance in a particular tissue you looked at between those two individuals, whether it's strains of organisms or people or whatever. But if you look across many members of a species, you're going to find that more than 50% of those transcripts significantly vary. And actually, now that we've got up to sample sizes of 30,000 is the latest large blood-based EQTL analysis, it actually turns out that we can see variants associated with the abundance of at least 90% of the transcripts in the genome. So that number there is probably more like 20%, and that one's probably more like 90%, if not 100%. So gene expression variability is extremely high, and it turns out that the genetic regulation of that has very high heritability. So in fact, estimates of heritability of transcription are in the similar sort of magnitude as visible traits. And that's not a priori obvious that it would be that way. I mean, you might think that the trait itself is just adding up all of these contributing variation in individual genes, and therefore that the heritability of individual genes might be quite a bit lower than that for visible traits. But that's not the case. What we now know is that the heritability of transcription is pretty much in the same order as traits. That is to say, if not, well, sometimes, occasionally it's up to 80 or 90%. More often it's in the range of 30 or 40% or 50%, which is not too different from obesity, diabetes, inflammatory bowel disease, and so forth. And if you ask the question, how much of that is due to many common variants, and how much is it due to a single major effect SNP, it turns out that one prominent E-SNP may have the largest effect, so it may explain like 60% of the heritability for expression of a particular gene. But typically multiple variants are going to explain that single SNP effect. So it's just capturing variation at different places. We'll see that a little bit later in the lecture. But also, if you actually want to know what's explaining all of the heritability of each transcript, then it turns out that that's due to cis variants and what we call transvariants. So it's ones elsewhere in the genome. And in fact, the cumulative effect of those transvariants, as we look further and further, is at least half, if not more in many cases, of the variants. So the bottom line from these two sentences is that actually the genetic regulation of transcription is just as complex as the genetic variation of traits when you think about the total contribution. But nevertheless, the power of E-SNP analysis or expression QTL analysis is that actually it turns out we can identify single variants or single sets of variants, even in sample sizes as small as 100 people, that are explaining as much as 5% or 10% of the variation in that single transcript. So there are large effects, and almost all of those are local, so they're in the vicinity of the transcript. So we'll see why in a second. Okay, so this is just a couple of examples. This is from peripheral blood gene expression from a sample of a couple of hundred bowel disease. And on the left here is just an example of a SNP, RS12627970. It's located in the vicinity of the SIN-GR1 gene. And what we're measuring here is the expression levels of the SIN-GR1 gene normalized to a mean of 0, okay? So if you look at the GG homozygotes at this SNP, their mean expression is actually a little bit more than twofold greater than the total sample mean. The heterozygotes is somewhat in the middle between that, so maybe 50% greater. And because there are many more homozygotes for the A allele, the major allele, their average is slightly less than 0, okay? So if you do a statistical test of that difference in gene expression, it turns out that the EQTL effect is significant at the level of P equals 10 to the minus 20, 10 to the minus 25. So unbelievably significant that, if our genome-wide association threshold is typically 10 to the minus 8, then this is many, many orders of magnitude greater than that. And you'll see it's true despite the fact that there's actually wide variability among individuals. Okay, and so the reason we have this power is because we've actually got a large sample size. Okay, so this is from RNA, just bulk RNA-seq data. Here's another SNP somewhere else in the genome, another locus somewhere else in the genome, where in this case, the GG allele actually has reduced expression. So the GG homozygotes are about twofold less than the heterozygotes, which are in turn about 2 or 1.5-fold less than the AA homozygotes. So that effect is in the opposite direction. This is a gene called DAP. And interestingly enough, in this case, both of these variants are the risk allele, okay? So what that's telling us in this case is that the risk allele is actually associated with increased expression of SYNGR1, and the risk allele here for GWAS is associated with decreased expression of DAP. And so just bear that in mind, because at the end of the lecture, I'm actually going to finish up with the concept of transcriptional risk score, which is actually adding up these effects and asking if they do a better job than the genotypes themselves of predicting traits. Just like a polygenic risk score, we can generate a transcriptional risk score. So the point here being that eSNPs can either increase or decrease expression, and if we want to relate that to the GWAS effects, then they can actually increase or decrease the risk of disease. Okay, so if we now go ahead and do this genome-wide and sort of take a snapshot of what's going on, two of the very first studies to do this on a genome-wide basis were actually in QTL mapping frameworks. So in other words, these weren't done in outbred strains of yeast or mice, they were done in a cross. Both of these studies were a cross between two common strains of yeast, or in this case, from Eric Schatz's group, between two strains of mice, in fact, an obese and a lean strain of mice. And they both gave very, very similar pictures. And that picture is that you could sort of map the marker location, so the SNP along the genome, against the trait location. So that in this case, the trait is the transcript. And you actually see that there's this big, long diagonal here, which is actually most of the density. It doesn't look like it, but that's actually most of the density. And if you actually home in, well, what it's telling you is that there's co-localization in genotype space. So in other words, the SNPs are in the same place in the genome as the variant it's regulating. And that's true for the vast majority of EQTL associations. However, against that background, you see these columns here of genes. So AMN1, LU2, this is actually yeast data from Josh Akey's paper. And what they are hotspots. They were called hotspots at that time. So places in the genome where, in fact, that single variant is not only affecting itself, but it's affecting many, many other targets. So that would fit the idea that the SNP regulates the transcript, and then that transcript goes and regulates all of these targets. At least that's one model of what could be happening. And people were very, very excited about this back then and thought, wow, if we could do this and it works in this genetic or genomic setting in the context of disease or in animal and plant breeding, that would be really awesome, because we'd actually just be able to map these major effect regulatory factors. And so these SNPs associated with those 10 or 15 major targets would probably be critical genes affecting complex traits. And all we need to do is to do that in a couple of hundred individuals rather than tens of thousands that it takes in GWAS. However, unfortunately, it turns out that actually each one of these variants is actually a lab-derived mutant. Okay, so these effects you probably wouldn't see if you took two wild-type strains of mice. So as time went on, it turned out that these were just variants that had been selected for artificially in lab culture without people realizing it. And so those major effects are actually would only be very, very rare alleles in natural populations, because they'd almost certainly be deleterious outside the lab context. Okay, so if you do this on 200 people or 200 outbred strains of mice or 200 lines of Arabidopsis, well, actually, I'll take that back. If you do it on animals, you won't see these columns of trans effects. you do see them more in plants, and we can debate why that is. It may be because some plants have more inbreeding, maybe domesticated species, which are what most plants we work with have been artificially selected like this. So you see a little bit more of it in plant in genetics, but in animal genetics, you tend not to see it. Unless you get to very, very large sample sizes, we'll see in a minute. But anyway, critical distinction is between cis-acting variants. So they're ones where the SNP is localizing to the same place in the genome as the transcript it regulates, and transacting variants, which are SNPs that regulate a gene somewhere else in the genome, usually on a different chromosome, but also maybe on a different part of the chromosome. So for example, down here, you might have a SNP. Well, actually, I can't. So here's chromosome 4, here's chromosome 4. So this SNP is acting here on itself, but it's also acting here on another SNP elsewhere on that chromosome. That would also be a trans effect. Heuristically, we typically define trans effects as more than a megabase away, but there's some debate in the literature about that. It's not that important to us today. Actually, formally, the term trans means on an opposite chromosome. So a trans effect is one from the maternally inherited chromosome to the paternally, that would also be trans. So over time, the terms cis and trans have been replaced by local and distal. And well, they haven't been replaced. They should have been replaced, but local and distal is a better term, but cis and trans are sort of stuck with us. Then the final point I'd make from this slide is if you can ask, well, how close does a SNP have to be to itself to be cis-acting? I said within a megabase, but the vast majority of them are actually within about 10 kilobases of the transcription start site. So large effect SNPs tend to co-localize with the promoter of the genes they're regulating. The majority do, but many, so maybe 30, 40%, are further away, and they're not necessarily acting on the closest promoter. There's also a general rule that ones that are located further away tend to be maybe more cell type specific, whereas ones closer to the promoter tend to be more universal in their effects on many tissues. But this is biology, so there's lots and lots of exceptions to that sort of generalization. All right. So I don't have a slide in here about power anymore, but because of the expense of doing this, so you've got to do both the genotypes of the organisms and the transcriptome. You're looking at something like $1,000 per individual. Classically, this has only been done on R01s, on relatively small grants. So you're looking at enough money to do maybe 1 or 200. So that's most of the studies. So what's starting to happen is you're getting large consortia pooling resources and getting to sample sizes of several thousand. So one of the first people to pioneer that was Ludo Franke and his group in Nijmegen in the Netherlands. And they put together something called the blood EQTL browser, which is the most comprehensive browser of EQTL effects for a single tissue, and that tissue happens to be blood for the unsurprising reason that it's actually very easy to sample blood, and it's used for a whole lot of purposes. So as a first approximation, or a first look, I should say, at genotypes, this is a really good browser people can go to. And this paper from Vestra et al. In 2013 from Ludi's group characterized EQTL by meta-analysis of 5,000 individuals. And then they replicated the findings in an independent data set of almost the same size. And so when they went, my very first paper in this field was actually looking at a study of 150 or so, I think it was, Moroccans. So I actually did a comparison of Berbers or Amazigh and Arabs living in Morocco between urban and rural environments. So I was very interested in G by E effects on EQTL. And that study identified about 100 significant SNPs, okay, and that was from a sample size of 150. And that's fairly typical for the field. So now we've gone up to 5,000, and you've got cis SNPs for almost half the genome, so 5,000 cis SNPs, and you even had 233 trans EQTL, so variants that at a genome-wide significance level, so 10 to the -8, are regulating, in this case, as many as 103 different loci. So one locus affected by variants in multiple different loci in the genome. And then many of those were disease associations. So this is a pretty comprehensive study, but actually was superseded last year by Umo Vosa, Nick Claringbold, and others. In fact, my own graduate student was the third author, I think, on that paper, from the EQTL Gen Consortium, where we did EQTL meta-analysis of almost 32,000 individuals. And they did that by combining data from 37 different cohorts. Okay, and this is all blood data. Some of it was microarrays, some of it was RNA-seq. In fact, most of it was microarrays, so less powerful than RNA-seq. But nevertheless, it's quite extraordinary that with genotype imputation, we were able to do 11 million SNPs with a minor allele frequency greater than 1%. Of those SNPs that we identified, 10,000 of them were associated with traits. So we were able to do some more further analyses of those. And we looked at almost all of the genes in the genome, the protein coding genes in the genome. That's 20,000 genes. So some of the findings that come out of this, was that cis-EQTL were identified for 88.2 or 17,000 of the genes in the genome in whole blood. And that's particularly remarkable because whole blood is itself a mixture of T cells, B cells, monocytes, macrophages, dendritic cells, neutrophils. Okay, so it's actually a very diverse collection of cell types, and yet nevertheless you're finding variants across all of that mixture and mixture of abundances that are having these cis effects. So that's quite extraordinary. 88% of the genes identified locally acting variants explaining some of the trait. In terms of trans-EQTL, those sort of bands we saw a couple of slides ago, we're now, with this sort of sample size, up to 6,500 sites in the genome having trans effects. So that's amazing. And and 1/3 of those are associated with traits from GWAS. Okay, so we'll look at that in the next slide, a little bit more detail. But whereas a cis SNP is locally acting, we're now saying that there are many thousands of cases where we're seeing trans effects at very, very high confidence and significance levels. And extraordinarily enough, actually replication across the 37 cohorts was actually larger for those trans effects than it was on average for the cis effects, probably because there's a bias to identify these ones. To get across the threshold, they have to be robust, more robust. And then Lourdes' group led something called EQTS analysis, where they actually said, well, let's take the polygenic risk score for a disease. So instead of taking local SNPs, we're just going to take the risk score for disease and ask how they associate with gene expression. So they defined this term expression quantitative trait score, and they found 13% of the genes were associated with some polygenic score for some disease. And a lot of the paper was actually about inferring consequences of that. And in fact, that's why this paper was actually out for review for about 3 years. It initially was sent to Nature and ended up in Nature Genetics. And I actually think it had something like 3,000 hits before it was published, because reviewers were upset about what I thought was some fairly minor components of the paper. So it's just one of those papers that I think is absolutely extraordinary achievement, not because we were involved, but because of Lourdes' group's efforts. But for whatever reason, it had a tough time getting published. But it's an absolutely extraordinary study, in my view, and lots in it. So I recommend you read it. But one of the things that I think is most interesting is that with this sort of power now, you can ask that question whether or not local SNPs are mediating trans effects through the gene they're regulating. Okay, so direct transcription factor regulation would be where you have a SNP mapping close to the promoter of a transcription factor. So they're associated with the expression levels of that transcription factor. And that transcription factor is known to bind in the vicinity of one of the trans targets, okay? So that's your simplest model for how an eSNP can work in trans. So it regulates locally its own transcription factor, which then goes on and regulates target genes. But it turns out that only a small fraction, so about 5 or 10% of the cases were of that variety. Much more common with these different types of indirect transcription factor regulation. So for example, you can have a SNP binding at one of these transcription factors, but that transcription factor itself does not have a binding site anywhere near target genes that the SNP acts on. So it's most likely that it's actually regulating the expression of some other gene, maybe a kinase or maybe an enzyme, something else, which in turn is then, or another transcription factor, which is then in turn regulating that downstream target. Or it could be regulating a non-transcription factor, which then mediates its effects through a different transcription factor, which binds the target. In fact, that mode of indirect regulation was the most common in the data set. So in fact, most of these cis-acting SNPs are not affecting transcription factors. They're affecting some other protein that then interacts with the transcription factor that mediates the downstream effects. And you can have other iterations of that. So these are the distribution. So about a third, no known mechanism, about a quarter this type, and then maybe a fifth of each of the others. And then there are a few other things there that you can read about in the paper. And then here's an example of one of these where, in fact, you've got a lot of this type of regulation. Okay, so this is a SNP in the REST transcription factor, neuronal specific silencer transcription factor. And it turns out that something like 28, I think, different targets of that all have binding sites in their vicinity for the REST transcription factor. Most of them downregulated, but some of them upregulated. Or I'm sorry, the other way around. Most of them upregulated, some of them downregulated. Okay. So expectation, you see a lot of this, and this one actually turns out to be also a GWAS hit for coronary artery disease. So it's helping to explain that mechanism. But more typical is having indirect regulation like this. Okay, but I hope you can sort of see the power and why there's so many groups interested in doing this sort of work, because we're now not just going from SNP to gene expression and then relating gene expression to the trait, but we're actually opening up the mechanisms by which these complex gene regulatory networks operate. Now, the limitation of doing it in whole blood, there's a couple of limitations, but one of them is that it's a mixture of cell types. So the very next step from this is to go ahead and do it on single cells. And Luda's group is now putting together a consortium of single cell people who will combine their efforts. There's actually, the data is not there yet, but the consortium is actively working, and they will probably have single cells from tens of thousands of individuals before long. But what we do have is this remarkable study from my colleague, Joe Powell, with whom I taught this module a couple of times. So, but he's in Brisbane, and COVID knocked out that. And happily enough, Tom has stepped in and giving us his wonderful perspective. But Joe's more like me. He's a stronger quantitative geneticist, but he's more an empiricist with a background in quantitative genetics rather than machine learning. So he was a postdoc with Peter Vischer, one of the leaders of the GWAS field, and initiated EQTL studies in Peter's lab. And I don't know how he did it, but somehow as a postdoc, he managed to put together a consortium and pay for this study of 1,000 people with about 1,000 PBMC from each one. So in fact, in total, he got 1.3 million peripheral blood immune cells. So if you remove the neutrophils and the red blood cells, you're left with B cells, T cells, monocytes, macrophages, and about 14 different types of immune cell. And he got them from 982 adult donors. So they're actually Australian adults in an eye disease cohort. And in analyzing this data set, he's now identified 26,500 cis EQTL and 990 trans EQTL. So that's a large number from 1,000. And I think you wouldn't see that in whole blood, but the reason you see it in his study is because these trans effects are much more uniform in single cell types. So this should get everybody hope that once we get below the tissue level, now to the single cell level, that these types of studies are going to be extremely revealing of biology across the board. Okay, so just so you can see this graphical abstract from his paper, but some points that are made in the paper. It turns out in his case that almost 20% of the EQTL localized with an immune disorder, GWAS association. So that's double the number that had been seen in, for example, IBD genetics, much larger bulk tissue analyses. And then he was able to go ahead and do this thing called Mendelian randomization to confirm that actually the genotype was mediating its effect on the trait through the expression of that transcript. So it's kind of like models like the ones I was showing in the previous slide. So direct effects. So that's just overnight, just explaining how like 4 or 5 times as many variants are mediating immune disease than we knew about previously. Second thing he identified, which I think is a really critical point in the paper, is that about a quarter or 20% of the ones he identified in B cells actually change their EQTL status as cells mature from naive to memory B cells. Okay, so that's this idea of context dependence. And as I said at the start, one of the reasons why this is 20%, not 100%, is that you have to look at different states. So in this case, the 2 different states were naive to memory, so immature to mature B cells. We're going to work with him in the next year or so to actually do even better annotation of double negative and switch memory and different subtypes of B cells and to get this more refined. But you know, that's a quarter of them a state specific, not cell type specific. And interestingly enough, two-thirds of those were identified as the interaction term in the model. Okay, so if you just looked in naive or just looked in memory, you wouldn't have seen them, and combined you wouldn't have seen them, but he actually identified them as being significant through the interaction term. So there's a change in effect size from naive to memory. I am aware of a colleague at the Sanger Institute, Kyle Anderson, who's doing similar studies, also in the context of autoimmune disease, but he's focusing on the myeloid lineage, I believe. And there, what he's doing is taking the cells ex vivo and treating them with different stimulants. So cytokines, TNF, and things like that. And just anecdotally, he's telling me that he sees this number going up. I think he said something like 70% of EQTL overlap to the GWAS associations when you look across multiple different activation states of cells. So it's not just maturation, it's actually activation states. I shouldn't have said that given that this is being recorded. So don't tweet about that. But it's a really important observation. Hopefully his paper will be out fairly soon. And there's in the literature, there's actually precedent for that at the bulk level. There's actually, even as much as 5 years ago, there were a couple of groups showed that in fact, if you treat cells ex vivo, you change the EQTL landscape in bulk RNA. An aspect of the paper that I actually don't really necessarily agree with Joseph about is that he claims that a majority of the effects are cell type specific, which is somewhat in contradiction to the bulk cell data. But what is clear that a lot of the common effects that are seen are concordant across cells. So something like 80% of the EQTL effects that you find in B cells are also of a similar magnitude in T cells. I think where we disagree is sort of in the term specific. So he would say that this difference from an effect size of maybe 1.5-fold is a specific effect relative to an effect size of 1.2-fold. I would call that a concordant effect. He would call that a specific effect. Okay, but so the bottom line there being that the magnitudes of effects also change across states. Okay, and then I think this is another one of those papers that actually was out on bioRxiv or medRxiv, whichever one it was, for 1.5 years before it was actually published. And the reason was he had to wait. I think the reviewers asked him for replication, and that was really difficult given that there's not a lot of single cell data, but he has managed to incorporate a replication in an Asian data set. And I think he's showing down here the replication rates are at least 70%, if not 95%, depending on the cell type that he's looking at there. Okay, but if that 5 to 20% really is ancestry specific, that has enormous implications, I think, for understanding immune disease, and it's also true of the nervous system and of liver and other tissues, at helping us to address disparities in genetic effects across ancestry groups. Okay? So I think we're only going to see more and more of this type of study. In fact, my own group is doing it in African Americans in blood, and with Pong, actually, we're part of a big study, the Gut Cell Atlas study, doing this in cell types, single cell types in colon, ileum, and rectal samples. And then the other study that's not single cell, this is just bulk, I shouldn't say just, but this is bulk data. So what the GTEx study is this amazing consortium study of the Genotype by Tissue Expression Project, where they're aiming to get close to 1,000 individuals at autopsy with RNA extracted from, well, at this point, 43 tissues. I think, I can't remember the exact final number, at least 50 tissues from all of those cadavers. And so the 2015 study had 175 individuals and a total of 1,600 samples. And so some of that data is shown here. The 2020 study was a series of 3 or 4 papers, follow up with 600 or so individuals. And if you go to the portal now, I think it's getting close to the 900 or so. So Kristen Ardley is the PI on this now, I believe. And so they're just doing an awesome job. So some findings of this GTEx project. So you might expect that the enrichment of EQTL is different across different tissues. Part of that is because of sample size effects. So what you're seeing here is the yield in terms of the number of significant E genes. So remember, an E gene is a target of an EQTL. So genes regulated in cysts. So the number of those is a function, a linear function almost, as a number of samples. Eventually it will plateau out. But so some of the tissues like blood have the most samples. But actually it turns out that blood is relatively unenriched. So the opposite of enriched compared to thyroid is the highest one here, the highest yield for any given sample size. So the slopes of those lines are significantly different. That might have to do with the cell type diversity within tissues, but it may have to do with just intrinsic aspects of cell tissue specific architecture. Okay. The second thing you can ask is how similar are different tissues? And as you might expect, tissues that are more similar developmentally tend to be more similar in their EQTL profiles. But what's clear is that every tissue has unique EQTL. So across the bottom here, so blood is the most divergent of all. And then this is muscle, heart. So muscle and heart are fairly similar. They're both muscles. Thyroid and artery and lung, sort of cardiovascular tissues. The nervous system tends to be out on its own. And then skin and adipose, somewhat more related. If you ask how many SNPs are unique to one tissue and how many are acting in many, many tissues. So this is enrichment for SNPs that act in single or multiple tissues. It turns out there is some fraction, and as I said, they tend to be promoter proximal, that are active in many, many tissues. I don't have my pointer on, sorry. Active in many tissues, those ones out here, and there are other ones that are only active in single tissues, okay? And then this final plot down here is actually saying, well, what about if we compare profiles between tissues within an individual for the same tissue between individuals and cross different tissue, different individuals, there's a gradient of effect. So if you look at the pairwise correlation, it's lowest for different tissue, different people, and it's by far the highest for the same tissue between any two people. So that's not really surprising, but it's nice to see it confirmed there. Okay, so I could go on for hours about the GTEx project. I won't, but I do recommend that you look not just at the 2015 papers, but Google it, and you'll find many, many, many follow-up papers from the GTEx consortium. I'm not going to talk in any detail about the software that's used for this. One of the standard tools for any sort of genome-wide association study is PLINK. It can also be used to do GWAS on not just one trait, but tens of thousands of traits, a little bit slow, but nevertheless that can be used. So PLINK is your basic workhorse for any sort of GWAS. It's good for EQTL. So Emil's asking, is there any info on which tissue is the most concordant with other tissues? I mean, it's certainly not blood. The brain tends to be very different from everything else, and all of the others share similarity. I don't think it's really a valid question. I think a more interesting question would be whether or not something like an induced pluripotent stem cell shares with more. And I don't know what the answer to that is actually. But certainly as cells differentiate, they gain their own profile. So it's not a simple question to answer. The fastest tool out there, and a very, very popular one, is something called matrix EQTL. So that's used for ultra-fast EQTL analysis. So it's the way it's coded and the matrices and things makes this an efficient software. So I recommend that. Gamma is one that we use a lot in my lab from Matthew Stevens' lab at University of Chicago. So it's called Genome-Wide Efficient Mixed Model Association. So I like that because it's in a mixed modeling framework. So you can include a lot of covariates. So it's actually really good for actually also doing powered analysis where you combine tissues, but actually evaluate tissue interaction effects. Or you can include, say, disease versus control. You can do male, female, also any sort of comparison. So it's a mixed model association tool. If you've got multiple samples per individual, you actually include individual as a random effect. I think you can include kinship matrices in Gamma. So that's sort of the more flexible framework. And then there are a bunch of Bayesian tools for particularly for doing fine mapping. I'll get to fine mapping in a minute, but DAP is the fastest of those. From the Wen lab at the University of Chicago, and another one out of UCLA is the Caviar Bayesian fine mapping tool. So these Bayesian methods are really optimal, not so much for genome-wide analysis, well, genome-wide fine mapping analysis, and I'll explain that in a second. Okay, in fact, I'll explain it right now. Okay, so if you go to the blood EQTL browser, or if you look up just about any GWAS database of anything, what they're reporting are typically univariate statistics. Okay, so what they're doing is they're walking along the genome and evaluating what the association is at any point in the genome, independent of all of the other SNPs in the vicinity. But we know that there's this thing called linked disequilibrium. So in other words, in the vicinity of any SNP, all other SNPs tend to be correlated in their allele frequencies and their allele distributions with that one because of the history of the way mutations accumulate in the gene pool. Okay, so within about 100 kilobases, in stretches of or haplotypes of about 100 kilobases, there's a high correlation structure to SNPs. Now it's assumed that EQTL effects are sparse, or it has been until recently assumed that EQTL effects are sparse. In other words, that there is really only one variant within a credible interval or within a haplotype block that's responsible for the effect. However, it's also quite possible that there are actually multiple SNPs responsible for that effect. And in fact, my own group adopts that as our basic framework, that we actually think, for a number of reasons, that actually most EQTL are not simple. So you don't reduce the effect to a single SNP, but rather they're complex. So there's 2 or 3 or more variants per locus responsible for the effect. And one of the reasons for that is you can do conditional analysis. So if I take a SNP and then I fit all the other variants around it with that SNP in the model, I still get residual effects in the vicinity that are explaining some of the variation. Okay, so that tells you right there that there must be a second SNP in the region. In fact, we've done this comprehensively in a set of 2,000 samples, a subset of that EQTL genome consortium. And it turns out that for every EQTL, about a third of them have a secondary EQTL that's in the vicinity, but is independent of it. And then a third of those have a third one. A third of those have a third one, pretty much. And so it goes down. And so there are loci where very clearly there are as many SNP, there's 10 SNPs in the region explaining its expression. Okay. So let's imagine then that this is a locus and that there's a gene somewhere in this region, say maybe here, which is being expressed, or actually anywhere along the locus, it's being expressed, and it's being acted upon by these variants. So what I've indicated that by here is imagining that there are variants that increase the expression, where the common variant increases expression or the major allele increases expression. And there are others where it decreases expression, okay? And suppose that the black lines are these univariate associations, then that's what you would get if there were maybe a distribution of actually truly acting SNPs. So the red ones, if we can find map them, would be the true causal variants, okay? So in this case, in this LD block, you might have two causal variants here and here, whose effects actually sum up. So there could be some other variant somewhere else in the genome that actually, not in the genome, in that locus, in that region of the genome, that actually sum up the two effects of those even better than each individual site does alone, just by chance, the way is the exact segregation pattern is. So in fact, what you conclude would be that that's the peak SNP. The parsimonious assumption is that that's the causal variant, but we now have pretty good evidence that very often it's just the peak SNP, it's the tagging SNP, but it's actually picking up effects at two other loci. And if they're operating in the same direction, well and good, then at least that's a really good tag for that effect. If they're operating in the opposite direction, so over here is another LD block. So we've got two SNPs. This one increases expression and that one decreases expression. They would actually cancel each other out, and you wouldn't see the effect of that locus at all. And we've got some papers sort of showing, just from a simulation perspective, that actually sometimes in this sort of scenario, you could actually induce another variant that should be genome-wide significant that would only is actually due to the effects, the combined effects, or what we call interaction effects. So the bottom line is that actually, because of the complexity of gene regulation, you don't expect to identify all causal variants at all in EQTL analysis. And in fact, that's probably true of GWAS as well. So to the extent that gene expression is regulated in these complex manners, we're now realizing that when you do fine mapping, it could easily be that many of these GWAS effects, and in this context, EQTL effects, are actually complex summations of the effects of multiple variants in links to equilibrium, and that the actual fine mapping of the causal variants is, from statistical methods alone, almost impossible. So one of my students, Emily, is actually using a crop-seq method to try to screen along regulatory regions for immune loci. And what we're actually finding is when we do that, in some cases, you find actually that the peak SNP is really the causal variant. And in other cases, we find really good evidence there are multiple variants in an LD block that have to be on the same haplotype, operating in the same direction. And so the major effect you see is actually due to the contributions of many contributing variants. Okay. So on the previous slide, I mentioned there are these Bayesian tools for doing that. So the problem, I mean, if you could fit models with every possible pairwise combination, you could, in theory, identify what the correct combination is. But in fact, there's hundreds of variants per locus. That's computationally infeasible. So these Bayesian tools adopt methods which sort of take into account the LD and do an empirical maximum likelihood estimation of what's most likely to explain the effects. Okay, so DAP and Caviar and some others are just tools for doing this fine mapping, and what they're doing is approximating the signal that's most likely to be responsible for these effects. And those tools are also suggesting that it's quite common that you've got 2 or 3 variants responsible for each effect. A few times I've used the term credible interval. So a credible interval is a set of all SNPs which could feasibly or plausibly explain the EQTL effect. Now, there's another tool which I think is really a critical and useful one, and that's something called CoLoC from Gianpaolo et al. From the UCLA group. And this is a Bayesian tool again, but it's a test for colocalization. This is a figure from their paper, which doesn't look like real data, but it actually explains the concept quite well. So there are so there's a null hypothesis, which is that there's no genetic association at that locus with either a trait or an EQTL. So what we're looking for is the co-localization of the SNP with the trait and the transcript. Okay, so the EQTL, GWAS, are they co-localizing? So H1 is the alternative hypothesis that there's only one EQTL at that locus. And H2 is the alternate, there's only a GWAS signal at the locus and they're independent, okay? So the two of interest, the two hypotheses of interest are H3 and H4. So H3 shown here is that there are two independent EQTL signals. So even though the GWAS hit and the EQTL hit are close to each other in the genome, H3 is that they're actually affecting different SNPs, that just because of the LD structure, they both give a significant result, but they're actually not the same SNP. H4 is a stronger hypothesis that in fact the effect is the same. And if they are the same, then the expectation is that the total profiles of the EQTL effects, univariate effects, should be very, very similar to each other, whereas in under H3, they'll be somewhat different. So my next slide shows a couple of examples of this. So on the left is, at the top, is the association at this FRK locus with low density lipoprotein levels. So this, so type of cholesterol levels. Okay, it's not cholesterol, but it's related to cholesterol. And so what? So the top plot is the LDL. The bottom plot is FRK. Okay. So if we look at this SNP 11,635,4539, it's the peak association here. It's p-value for GWAS. It's GWAS significant 10 to the minus 8 for LDL, and it's actually this SNP here, and so it's also significant 10 to the minus 12 for expression of FRK. But if you look in detail, the profiles of those actually don't look the same, right? So this block of SNPs here around that peak actually is this block here, the blue block there, and that peak here, that's the peak for FRK, actually happens to be these green ones here, okay? So they're actually independent associations, even though they're very close together, and they're somehow picking up on each other's signature enough to be significant in both analyses. So if you're just naive about it and look up a database like bloody EQTL and you say, oh, here's my GWAS hit, here's my EQTL hit, they're the same thing. Well, actually, according to this analysis, they're not the same thing at all. They're the same locus, but they're a different variant, okay? On the right-hand side, it will be a situation where actually you get H4 supported. So this is the gene SDC1, and then this is total cholesterol in this case. And it turns out that that's actually not genome-wide significant, but this one is genome-wide significant, okay? And we've got posterior probability. This Bayesian model is 82%. And so it's actually saying that it's very, very likely. In fact, the best fit model actually says that that peak there, these red ones in high LD with 203.71380 is the same peak as this one in high LD with 203.868519. So those two SNPs there are probably just a few hundred base pairs apart in extremely high LD, which is what the red color indicates. And so even though a different SNP is the actual peak, one or more of those ones underneath that peak are responsible for both of those signals, okay? So that's very powerful because it allows us to now go ahead and say, well, is this EQTL really responsible for the trait? And in this case, it wouldn't be. There's another method called summary Mendelian randomization from John Yang and Peter Vischer, which has gained a lot of popularity. And it's much more just a simple correlation of, that's not what it is. There's more theory behind it, but I don't want to explain the theory in the few minutes we've got here. But it's basically, in the end, turns out to be somewhat equivalent to just looking at variants that have both a strong EQTL effect and GWAS effect. So you can see that there's some variants under this SMR model that appear to only have the GWAS association, some that only have the EQTL association, but there are others that have a very strong both. And so these red ones, these dark red ones are one, and the light red ones are ones that are significant by the SMR model as being co-localized. if we map those ones onto the colloque in our own study of blood EQTL variants, it turns out that, so this is the SMR significance value, that some of the ones supported by SMR are supported by H3, and some supported by H4, okay? So, two different methods, two different strategies for co-localization, giving different results. Personally, I kind of trust the colloque better, but I do have to say that colloque is a function of sample size and strength of effects, and there are reasons why it may be misleading in some cases as well. Anyway, the good news is you've got methods for evaluating this. So limitations of co-localization analyses, well, it's heavily dependent on statistical power and contributing analyses, which is generally relatively low. So if you're only dealing with a sample of a couple of hundred, then you're probably not going to get large enough effects from the EQTL to do this. The imputation quality of the SNPs is important. So actually going to a question we had on the previous lecture, this is one place where imputation can be actually very, very useful. So this is where you're guessing what the SNP type is based on the LD patterns rather than directly typing it. You're assuming when you're doing this analysis, the GWAS and EQTL are evaluated in the same population. In other words, there's no stratification across ancestry groups. You can get negative results if you're actually not comparing the right tissue or tissue state. And a lot of the Bayesian methods actually assume that there's a single causal variant. Well, I'm sorry, that's not true. The analysis assumes that there's a single causal variant responsible for each independent peak. Now, on the right here, I'm showing an example of a situation where you can actually overcome some of these issues. So this is the peak association for LDL. This is the peak association for expression of SYPL2. P posterior probability for hypothesis 4 is less than 1%. So in other words, this peak and that peak are not the same. What we did here is we went ahead and said, okay, well, let's just condition all of these other SNPs on that peak. And when you do that, you're left with a residual signal, still at 10 to the minus 15. Look at this, this is 10 to the minus 100. So that's maybe explaining 20, 30% of the variance in that transcript. This one's still explaining 5% of it. It's independently significant. And it actually happens to be not in LD with the rest of them. That's why it's retained. So it's these blue ones here, and those actually co-localize to the LDL peak. And so actually H4 is extremely highly supported for the conditional analysis of that variant, okay? So we can get clever and go ahead and do all sorts of things like that. And so the take home from this is actually oftentimes it's now turning out, so maybe 20% of the time, the peak association in EQTL analysis is not the one that's the GWAS variant, but actually it's the secondary or tertiary variant that's the ones associated with the trait. Here are some tools for doing these things. So Matthew Stevens' lab is the one that's developed this joint mapping framework in a Bayesian context. So that's really, really useful. This is Gemma. Roger Pique Reggie's group is the one that developed some of these alternate Bayesian fine mapping methods. And then his student, I can't remember what his name, SISG2022, I think, developed this DAP fast encoding of their Bayesian software. So there's some tools here that I recommend highly for doing fine mapping of multiple causal variants at a single locus. So I'm at 3, so I'm going to try to rush through in about 15 minutes some other results you can get from EQTL analysis. So this is one from Joe Pickrell when he was a graduate student at the University of Chicago. Really quite extraordinary result explaining why you see additivity of gene expression. Okay, so what they did here is these are all the pileups, the read counts for each of the exons in this gene model at this locus, okay? And these are the number of reads you get on chromosomes that are homozygous for the G, heterozygous, or homozygous for the A. So this is just a really nice RNA-seq visual representation of a classical EQTL. So high abundance in the homozygotes, intermediate abundance in the heterozygotes, and low abundance, almost no expression in this case whatsoever in the alternate allele, the rare AA homozygotes. And look at this. This is a small sample of just 100 individuals. Okay. Now, the other thing they did at that locus was just go in and say, well, let's look at these 35 GA heterozygotes and actually look in the coding region. So within one of these exons, there actually turns out there's a SNP, and they can ask what's the ratio of read counts between the G allele and the A allele in the RNA-seq pileup. And it turns out that if you do this across many loci, there's a really nice correlation, strong correlation, highly significant correlation between the ratio of the G to A, and the magnitude of the difference between the two homozygote classes in the total population. So in other words, allele-specific expression matches to EQTL effects that you observe in total read abundance. And that actually helps to explain why gene expression effects are additive. So it turns out that the polymorphisms affecting each allele are somewhat independent of each other. So if you've got a variant in a transcription factor binding site on this AA homozygote, you almost get no reads. Whereas if you have a variant on the G allele, you get sort of, if you've got one G allele, you get that much reads. If you've got two, you get twice as many again. So it turns out that this type of analysis is telling us that actually there's a molecular basis to the additivity of genetic effects at the gene expression level. Jonathan Pritchard, who is the PI of this, of Joe's at the time, has now moved to Stanford, has come out with this WASP approach. I think this is Jonathan's. So they actually argue that actually you're more efficient at mapping EQTL if you actually, they use something called the combined haplotype test. So they're actually jointly modeling the effects of the allele-specific expression, this imbalance, and the total read depth, so the classical EQTL effects. And so they actually have some pretty good compelling data that WASP is a better tool, a more sensitive tool for finding EQTL, although that was 5 years ago, 7 years ago now, and it hasn't taken off as much as I would have expected. So, not all variation at the transcript abundance level is due to transcription. Quite a lot of it turns out, is due to splicing. So here's a nice example of that. So it's similar to what we saw before. Here are the exons in green, and then there's an intron here in black, and it turns out that there's a variant in this case that affects whether or not there's inclusion of this alternate start site for the exon, for this exon here. So in the AA allele, there's a disrupted 3' splice site, which means you get inclusion of this ontronic sequence. And you'd be surprised, but actually it turns out that many associations with disease of this form, where the splicing EQTL is responsible for production of an aberrant protein, in many cases introducing a reading frame disruption, so you don't even get a proper functional product out of this. So more and more people are looking at splicing QTL as well as expression QTL. Splicing comes in different types of forms. So there's a bunch of software out there for looking at splicing. So this is software called Meso in the Burge lab at MIT. And it's just pointing out that there are lots and lots of ways you can affect splicing. So one is having alternate first exons. So many genes have two alternate promoters. This is a case where you have a mutually exclusive exon. So exon 1 is included in some transcripts, and exon 2 in another exon. Well, this would be exon 1. So this is exon 2, this would be exon 3. You get one or the other. So that's mutually exclusive. You can also get situations where you completely jump over an exon. So in this case, exon 6 is excluded from some transcripts. You can include introns sometimes. You can get alternative donor splice sites, alternative acceptor splice sites, and you can even get alternative 3' ends of transcripts. So if you go in and really look, so you don't get any of this sort of information from single cell. You can only get this from bulk. And in fact, to get it in a high lot of detail, you need to do targeted bulk sequencing. So Kira Berger in my group just graduated, and her thesis is about using targeted RNA-seq to characterize splicing defects in clinical cases of early onset disease. And so, and then I've skipped out some slides here, we're getting on, but it turns out you can also do the equivalent of EQTL analysis to look at methylation profiles. Okay, so there the trait is the transcript abundance, it's the level of methylation. You can do it on protein levels. So there are now methods using aptamers to capture proteins throughout the genome. There are other methods of doing proteomics. So there's protein QTL. I think I mentioned there are chromatin conformation QTL. So you can find variants that affect whether or not two regions of the genome make contact with each other by that high C assay. There are ATAC-seq QTL. So you can find variants that affect the openness of the chromatin. And putting those together with the EQTL or the methylation QTL allows you to put together very, very powerfully integrative models of what it is that regulates and explains variation in transcript abundance. I just wanted to finish up with just one result from my group that illustrates one of the things we're interested in. So this is Urko Marigota, and I published a review in 2015, sort of arguing for this concept of a transcriptional risk score. And then it followed up showing that it works in inflammatory bowel disease a few years later. So the idea is this, that we want to understand how it is that variants impact the trait. And so we get two types of information. So one is just a straight EQTL information. Okay, so if you go back to the very first slide I showed you in this lecture. So this is a case, very, very typical EQTL profile where you have AA homozygotes, less expression than heterozygotes, less expression than the BB homozygotes. And what you're doing classically in GWAS is basically saying, well, this is the risk. This is the risk variant. So individuals with this combination are more susceptible to disease than individuals with a protective variant, and then heterozygotes halfway in between. Well, if you think about it, and what's going on is that this variant is mediating disease through gene expression, then actually the individuals most at risk of developing disease aren't all of these. They're actually the red ones here. Or it doesn't have to be a threshold, but just the idea would be that the ones with high expression will be the ones most at risk. And the reason why the BBs are more susceptible is because they're more likely to have high expression. Okay. So the argument then is, what if we just add up, instead of the number of genotypes that you carry and generate a poly, which is what you do to make a polygenic risk score, what if we add up the average gene expression? And we have to do a few other things to make that work. So one is we have to recognize that sometimes the susceptible allele has reduced expression. So we polarize the risk and then we add it up. And so in this particular review paper, we just simulated this sort of scenario and said, well, yes, we expect genetic risk scores to be correlated with transcriptional risk scores, but under and then this is just showing that the odds ratios that we simulated were similar to what you see in GWAS. And then this is just the area under the curve analysis, sort of showing that actually, in theory, the transcriptional risk score should easily outperform the genotypic or the genetic risk score. So you get higher sensitivity with higher specificity with the transcriptional risk because it's closer to the actual phenotype. And so we went on and actually did this in the context of Crohn's disease. And so this is a result. So if you look at genetic risk score in a small sample, so this is just about 200 patients compared with only about 30 or so controls. And the reason the control number is so low in this case is because it's hard to get biopsies from children, from pediatric cases. But on the genetic risk score, not surprisingly, there's no difference because genetic risk scores just aren't that discriminatory with small sample sizes. But in the same scenario, with the same set of genes, in this case, it's about 30 or 40 genes, the TRS concept gave almost, well, not perfect discrimination, but all of the controls were below the mean of the transcriptional risk score. So the p-value on that discrimination was 10 to the minus 13. And then unexpectedly, but delightedly for us, it actually turns out that these genetic risk scores provide no discrimination whatsoever for disease progression. So B2 and B3 are progression to very severe stricturing or penetrating disease. Well, it's maybe not, it's not clear if it's clinically useful, but nevertheless, we've got a highly significant discrimination by the TRS with this measure between the progressors and the non-progressors. in this situation, despite a fairly small sample size. And we've now gone on and just last year published a study doing the same thing in ulcerative colitis, and actually then showing that we can actually predict gene expression and do so look at transcriptional risks derived from predicted gene expression, and several other groups are starting to do the same thing. And it turns out that actually these predicted gene expression risk scores also outperform the genetic risk score. So that's sort of combining TWAS with polygenic score analysis. So that's just an example of the way we're going. And I think when we get this to the single cell level, we're going to be even more powerful, that we'll be able to go in and for individuals through your Paneth cells, whereas somebody else's genetic risk is being mediated through their B cells or whatever it is, and really use that as a mode to sort of go in and advise on likely clinical strategies. Okay, I think that's all I wanted to talk about in this lecture. I'm happy to stop and answer any questions about this or my other material. Yes, I have one question there.